Hi everybody, this is Barot. I'm a PhD student at NYU Center for Data Science, and today I'm going to be talking about automated protein function description for novel class discovery. So protein function prediction, as it is usually framed, is a supervised multi-label problem where sequences are mapped to labels organized into a hierarchy, such as the gene ontology. What this project is centered around, though, is a new paradigm, which is that given a set of proteins, describe their common function in natural language. So why do we do this? Why do we make a model that describes common functions of a set of proteins in natural language? So first, why use sets of proteins? A function description is our abstraction of the common property of a group of proteins. So we discover functions by understanding that a group of proteins do something in common. And so why use natural language? We can avoid having our predictions be limited to a predefined set of functions, and with language, the model can compose new functions out of the same pieces that we use to explain the world to each other. In order to discover new categories of protein functions, with info to guide experimental design as well to test for them, we need a model that generates functional descriptions in natural language. So currently the methods for function prediction exist, but there's not that many that consider function discovery. Clustering-based methods that just tell you that a group of proteins does something different from others doesn't really give you that much information to test for those new functional categories. Deep Goes Zero is an example of a zero-shot learning method for protein function prediction, but it relies on ontological relations with known terms, so it's a little bit less flexible than just having a natural language description as the output. So the proposed model that we have is a transformer encoder-decoder type of model that is similar to those used in natural language translation tasks, where in this case we're using a set of protein sequences as the input, and we then use a length transform in order to convert that set of abstract feature representations into the same length for each protein. And then we average pool these sequence representations, and then we use a transformer decoder in order to actually get the natural language output. And so the model is estimating this probability of a description given the sequence set. And if the sequence set is more specific, that there's like a subset of the sequences in the set, it should give a more specific output. So in order to evaluate our model, the generated descriptions are pretty difficult to evaluate. So we choose to evaluate the probability scores that the model assigns to pairs of sequence sets and their known descriptions. So one of the metrics that we're using in order to evaluate the rankings of these scores is correctness, which is the average number of times the correct Go term outranks an incorrect one for a given sequence set prediction. The second metric is specificity, which is the among correct Go terms, the average number of times a child term outranks its parent term. And robustness, which is the average rank correlation between the predictions of a pair of different but identically annotated sequence sets. So the data that we're using in this is the UniProt KB SwissProt database, which is manually annotated and reviewed. And we use a minimum set of number of proteins and maximum number of proteins to restrict the Go terms that we're interested in. And we have 4 different sets of proteins and functions. So we have training proteins and training functions, which the model has been exposed to, and we have these sets of proteins for which there are some test functions that the model is not exposed to, and vice versa for test proteins and training functions, and finally test proteins and test functions. And so the model performances we show here in our table where we can see that test proteins and functions are slightly less given correct rankings by the model, and specificity overall is fairly consistent, and robustness is much better for those training proteins that have actually been exposed to the model rather than these two categories of test proteins. So with that, there's some test generation examples, which we take a set of proteins from the test set and try to generate a description using beam search. So this actual description of this sequence set and the prediction is pretty similar, except for that the prediction is a little bit more generic. In this one, the prediction is of a different organ than the actual description is. In this one, the prediction is fairly different from the actual description, but both mention DNA. And in this one, the prediction is more specific than the actual description, which may or may not be correct. This is talking about directed movement from the endoplasmic reticulum to the nucleus. With that, I would like to thank my collaborators, Vlad Gligorovic, Rich Bonneau, and Kyungyun Cho from NYU. And Vlad is from Genentech. And you can contact me at meetbarot@nyu.edu. And here are the references.